Okay, today's speaker is Chris Emerling, a new postdoc in the MVZ who's on he's working in my lab and he's an NSF postdoctoral fellowship. He came from UC Riverside where he did his PhD with Mark Springer, and he's very interested in xenarthran evolution. I have some fun facts here. You say you've only had 5 days in your life without owning a dachshund.
They're sad days.
So I guess you were a mammologist, except for from birth or something. And it looks like you like music from the 60s and 70s. And the title of this talk is right here, so I'll let Chris take it away.
All right, thanks, Michael. Can someone get the lights a little bit back there? Just to get it on the front right here. I just want to thank you guys all for attending. So far, it's been really great to get to know each and every one of you that I have, and yet I'm sure I've only gotten through maybe a quarter of the museum so far. So by all means, if I haven't met you, introduce yourself. I'll do my best to introduce myself to you as well. But so far, you've all been very friendly and very intelligent people contributing different ideas I've had. So. Anyway,
if I can just interrupt, I also like to say where the new people are sitting. I don't know if I said that. So Chris is in the office behind Rory's office on this side of the museum with Noel and others back there. So that's where you can.
Yeah, so come harass me. Anyway, so yeah, I'm going to talk about the regression of visual genes in various mammals that have adapted to subterranean environments. So the eye, as you can imagine, is a complex organ that has many different features that contribute to sensory perception of the environment. And generally speaking, if you look at mammals, the eye is pretty darn conserved. There's always some modifications that allow for adaptations for dim light and bright light. But as a general whole, it typically is preserved as it is. But what we would imagine ultimately is that there will be some regression that might be happening at the genomic level. So even if you're not seeing a lot of these morphological features, there might be some genetic components that are lost over time. And sometimes that does correspond with morphological regression as well. So when I was doing this work, I was kind of overall interested in this idea of how extensively does the mammalian visual system regress, period. So there hadn't been a lot of work done in terms of genetics looking at this. We knew about a few genes here and there that seemed to be inactivated over time, but not in terms of kind of big comparisons across the genome. And this might seem obvious, but the ultimate question is, does this regression correspond with light availability? So in bright light conditions, you might imagine that most of these genes will be preserved. You want to retain some of those same functions, but under dim light conditions, perhaps some of them are lost over time. And so these are kind of the broad questions I was interested in while doing this work. And so the way we typically look at visual regression, or just genomic regression as a whole, you can look at gene loss entirely, and that always gets a little tricky because with genome assemblies, you're uncertain whether or not was the gene lost or perhaps this wasn't assembled correctly. But when you actually are able to find unitary pseudogenes, so genes that have only a single copy, and that single copy has degraded over time via the accumulation of various inactivating mutations, such as premature stop codons or frame shift indels. And in terms of thinking about how the visual system might regress in a different species, looking at subterranean mammals seem like a very good system to actually study this potential for this phenomenon. And so subterranean mammals, they actually vary in their commitment to being underground, but there are some species that spend almost entirely their entire lives underground, only occasionally coming out perhaps for dispersal every once in a while, or maybe if their burrows have been broken. So before I actually get into the details of the studies, I'm actually doing a real basic biology lecture. You guys probably all know this, you're all obviously a very intelligent crowd, but nonetheless I want to kind of get you on the same page of the difference between rods and cones, because that'll come up a few times during this talk. So rods and cones are different photoreceptive cells that are present in the retina of vertebrates. They are distinct partially based off their overall morphology, so cones have a sort of cone-shaped outer segment, and so outer being further out from the center of the eye, whereas rods have this sort of rod-like outer segment, very convenient. These colors are actually flipped. This is a cone right here, these are rods. And they're different in terms of their morphology. But they basically do the same thing at the molecular level. They have paralogous pathways that are performing basically the same function of absorbing light, converting that and transducing that into an electrical signal, which sends electrical signals to the brain ultimately. But they're different in how they actually do this. And I won't go into the details why. You can look at some of these examples here. But generally speaking, what you need to know is rods are more sensitive. They're very useful in dim light conditions. Cones are a lot less sensitive, but they're very useful in bright light conditions. So even though the lights have been dimmed a bit here, overwhelmingly you're still using your cones. If you ever want an idea of when you're starting to use your rods, I always like to think of the example of walking into a movie theater. So you just kind of got some popcorn, got a soda, and then you walk in, and then you go, oh crap, I don't know where the hell my seat is, okay? That moment right there is you've just switched from going to cones to switching over to your rods instead. And in the bright light conditions, they become completely oversaturated. And so the signals aren't actually transducing anything to your brain. And so what happens is they have to slowly adapt. And you'll notice as you get into a movie theater or you're walking inside the house at night, you'll very slowly adapt. Okay? So that's kind of giving you an idea of the difference in the function. And so you can imagine under dim light conditions, rods become more important. Bright light conditions, cones become more important. Right? So getting to the actual research I did. Since subterranean mammals provide a nice example of independent acquisitions of becoming subterranean and living in these very dim light niches, we would imagine that they provide independent acquisitions or independent tests of this hypothesis that living in dim light conditions might regress your visual genome. And so for my first study here I'll talk about, I looked at these three different species. This is the Cape golden mole, Chrysochloris asiatica. This is the very bizarre and familiar star-nosed mole, Condyloma cristata. And this is the very ugly but very interesting naked mole rat, Heterocephalus glaber. And so these three species, they've had their genome sequenced, and they represent completely independent acquisitions of the subterranean lifestyle. So they're actually so divergent from each other. Their most recent common ancestor for some of these is about 80 million years ago. So even though you might look at this guy and think, oh, they're both moles, and so aren't these monophyletic? These guys right here are actually more closely related to things like elephants and manatees. So it's a completely different acquisition. But once again, it allows for independent tests of this idea that when you live in dim light niches, does your genome actually regress? And so we would imagine there would be some regression of the genomes, but in terms of the visual genes, but we would think that there's probably still retention of some kind of visual photoreception. So even when you live in a completely dim light niche, it's not that you never encounter light ever at any point in time, right? So if you're subterranean, once again, you might get a break in your burrow. And so by actually being able to detect that light, at least to some extent, might be useful, right? But otherwise, overall, we might not expect that most of these genes are retained. But we might expect that there be a difference in which genes are lost over time. Since cone-specific genes are mostly used in bright light conditions, we expect that cone-specific genes would be lost preferentially, whereas rod-specific genes would not, as well as certain shared genes. So there's some genes that are expressed in both rods and cones, and we expect since it's keeping a function for rods, it would retain those as well. Some exceptions might exist, particularly functionally redundant genes. So you can imagine some genes that have become duplicated over time that perform essentially the same function, maybe a little bit different, but they might be useful in bright light conditions. Having an extra copy of that gene could be particularly useful. And so if there's any functionally redundant genes, perhaps one copy is lost while another is retained. The other hypothesis I was interested in testing here is that vision genes are actually inactivated post-subterranean adaptations. So could we actually tie the loss of these genes to the invasion of a subterranean niche? And that might seem very obvious, but we do know to some extent nocturnal species will have some level of genomic regression that happens in vision genes. So perhaps it's not necessarily being subterranean, but it's just living in any kind of dim light niche. So being able to actually tie this to a subterranean habitat would allow us to test that hypothesis more thoroughly. So I looked at these genomes, looked at multiple genes. I'm not going to talk about all these genes, obviously. But we looked at multiple outgroup comparisons for each of the species and examined them for various inactivating mutations. And we used a method to actually date the inactivation of genes. So we actually used this sort of molecular clock method to date when these genes might have been inactivated. So the first thing I'll talk about here is kind of reiterating that first hypothesis that we were testing. We would expect cone-specific genes to be lost more likely than either rod-specific or shared genes. So right here, these turquoise triangles, these are cone-specific genes. These blue squares are shared, so they're expressed in both cones and rods. And these black circles right here, these are rod-specific genes. Okay? And what these lines are basically showing are interactions that have been demonstrated experimentally, or at least inferred based off of chronology. But generally speaking, what's most important for you guys to see here is the kind of cone versus rod specificity here. Okay? Now this isn't all 65 genes, I want to point that out. There's other things going on, but I'm just going to focus on this aspect right here. So these red X's I have superimposed, what those are showing are any genes that have been inactivated in one or more of the subterranean species. Okay? And what you'll notice from the get-go here is these cone-specific genes have been lost in quite a bit of abundance. And they've actually, in some cases, been lost repeatedly in subterranean lineages. So once again, we would expect that to happen. The rod-specific genes, however, are almost entirely intact with one interesting little exception. This is an ion exchanger, and so what happens during phototransduction, ultimately some ion channels are closed, leading to hyperpolarization of the cell. And so this gene in rods is specifically involved with rebalancing those ions. And so we know in humans that when this gene is knocked out, it leads to night blindness, so rod dysfunction. So it was kind of interesting that we found this knocked out in a few different species, but no other rod-specific genes were. One possibility is perhaps there's a splice variant that's retained here, and maybe the inactivating mutations we saw are somehow bypassed by different splice variants. There's also a cone paralog right here that performs the same function in cones, but it's also expressed in the brain as well as in retinal ganglion cells. And so it's possible that that's been co-opted for rod function, but that's a hypothesis to be tested for another day. But generally speaking, rod function has been retained. And then in shared genes, you'll notice they've mostly been retained too, with two exceptions here. GUCI2F and GUCA1B. You'll notice GUCI2F has a paralog here, GUCI2D. GUCA1B has a paralog at GUCA1A. And we know from functional studies, as well as disease studies in humans, that these genes are basically functionally redundant. And so GUCA1A and 2D are probably able to carry out the same functions under dim light conditions. So once again, this seems to support our hypothesis that cone-specific genes are lost preferentially, not so much rod, and only shared genes are the ones with shared functions. Now, one interesting little tidbit that I discovered when doing this research is that golden moles lack cones entirely. Now, this might sound like a common thing. Perhaps a lot of nocturnal species lose cones. Perhaps a lot of subterranean species lose cones. But in fact, when I discovered this, that was the first example of a terrestrial mammal to have been confirmed through genetic or molecular methods to lack cones entirely. And just the year before that, we discovered that in some species of deep diving whales, it was the first example of mammals that have lost cones entirely. It's actually quite a rare phenomenon. I'll talk about another example in a moment here, but it's pretty rare. And it seems to be that if you live in an extreme dim light environment, you lose cones. But being nocturnal is not enough. Bats still have cones. A lot of subterranean mammals have cones. So tucos, for example, those still have cones. Okay? So it is rare, but it seems to be suggestive of the extreme adaptation commitment that golden moles have to living underground. And if you actually look at golden moles, their eyes are completely covered with skin and fur. And so once again, it would suggest that perhaps the idea of retaining cones is not particularly important for these species. So getting back at the next hypothesis, so I wanted to test whether or not vision genes are inactivated post-subterranean adaptation. I'll give you an overview based off of the golden mole example here, because it had the most genes actually knocked out, so it's a good example here. So what this is showing is sort of a diagram of a phylogeny. I'll explain what's going on here. So here's Chrysochloris. This is the genome that's already sequenced. And then here's Amblysomus. Amblysomus hottentotus is another golden mole. And basically, their split probably indexes the crown golden mole group It's not entirely sure, but we think they radiated about 12 million years ago or so. And we were actually able to confirm in this species that they also lack cones. So it seems to be probably something that was lost in their common ancestor. And then when we go back a little, what this is showing here, this is a fossil species. So this axis actually responds to time. So fossil species is another fossil species. And this is going all the way back to the early Eocene. Okay? So for ancestral state reconstruction, all golden moles are subterranean. So you could probably put the adaptation of the actual acquisition of being subterranean right here at the common ancestor. But the fossil record actually is a little more informative than that. So this species right here, Prochrysochlorus myascenicus, there's fossils of these guys where you have the skulls, and the skulls are actually sort of shovel-shaped. And the way golden moles dig, they don't dig the way that, say, a star-nosed mole does. They don't use their arms just like this in a swinging motion. What they do is they get their skulls kind of in the dirt there, and they get their paws up here, and they kind of push up with their skulls like a shovel, and they push down with their forelimbs. Okay? So these modifications in the species that have a shovel-like skull suggest that becoming subterranean probably dates back at least to this species, which at its earliest was about 23 million years ago in the early Miocene. Beyond that, we have Eochrysochlorus, which dates to the early Oligocene. The thing about Eochrysochlorus is it's just a jaw fragment, so we don't really have any evidence of subterranean modifications. So at the oldest, we can index that perhaps becoming subterranean golden moles dates back to, at most, 23 million years, based on our available data. So these black branches here suggest not being subterranean. The brown is basically inferred subterranean acquisitions. Now these individual lines on these branches, these correspond to estimates for gene inactivation, so vision gene inactivations. And what you'll notice is, over this entire time period, so this is about 30 million years time right here, we only estimated 3 genes being knocked out, and it's quite spread out, if you can see that. Then when you get to basically the appearance of Prochrysochlorus in the fossil record, boom, 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 boom. You have all of these gene inactivations in succession happening right there. In fact, the first gene associated with with cone loss, GNAT2, happens right around there. So suggesting that cone loss seemed to have happened when these species became subterranean. So we did that for all 3 subterranean species. We actually also included some data. At the time, there wasn't a blind mole rat genome published, so one day I'll be reanalyzing this. But there were 2 visual pseudogenes that we had available, and the marsupial mole also has a single visual pseudogene present. And so out of all of these genes, the number that post-dated the inferred subterranean entrance, 30 out of 35 were post-dating, suggesting that once again that becoming subterranean specifically seems to be associated with this massive visual gene loss in these various species. So, given all of this stuff that I learned while working on subterranean mammals, I didn't really expect this to happen, but I'm very interested in xenarthrans, and actually that's why I'm here to work on xenarthran genomics and learn about a lot of different aspects of their biology at the genomic level. But xenarthrans hadn't really been studied in terms of their vision, at least not very much, and certainly sporadically, definitely not at the genetic or molecular level. And there's a good reason for that if you actually just spend any time thinking about the ecology of these species. Armadillos, they do dig, some of them burrow. I will talk about one highly subterranean or 2 highly subterranean species, but a lot of them are out above ground during the day, during night. Anteaters, some of them live in trees, some of them are on the ground, day, night. Sloths are living up in the trees. They're not living in these extreme dim light habitats. They're not they're mostly not very colorful. This is one exception. This is a male 3-toed sloth of a specific species. But generally speaking, not very colorful. So people haven't really looked at their vision and thinking, okay, maybe their vision's interesting. I didn't particularly think they would be either. But I spent a little time looking into this. And I started coming across fun little quotes like this, suggesting that, at least in armadillos, perhaps their vision is particularly bad. If you finish reading that, it's kind of a funny little commentary. And then sloths as well. There are these little anecdotes that kind of suggest that not only is their vision bad, but it's like really, really bad. Like they are like dumb blind, right? And if you kind of search the literature, you have to scour it. you'll see these little tiny studies here and there that suggest maybe they lack cones entirely. In fact, their behavior would seem to suggest that. When you lack cones, once again, thinking about that movie theater example I gave you guys, you walk into that movie theater, initially you can't see anything. So if you're in bright light conditions, let's say the tropics, losing cones seems like a really bizarre trait. So, but either way, looking at these different little commentaries here and there kind of seem to suggest that maybe there was something going on with their vision. And specifically, I wanted to test the hypothesis that this group, so Xenarthrans are, once again, armadillos, sloths, and anteaters. I wanted to test this hypothesis that they were rod monochromats. Seems very unlikely, but nonetheless, it seemed possible based off of their description. And because it seemed like some reports from sloths and armadillos, not so many on anteaters, unfortunately, because they were suggestive of this particularly degraded vision, that might suggest that the loss of cones happened in their most recent common ancestors. So that was another hypothesis that I wanted to test. So at the time I worked on this project, there were only genomes for 3 different Xenarthrans. Dasypus novemcinctus, one of the 9-banded armadillos, Colopus hoffmanni, one of the 2-toed sloths. And there is also a very fragmentary but still useful genome of an extinct ground sloth species, Myelodon darwinii. So we looked at this, these 3 different genomes, and we focused only on genes related to cone and rod phototransduction. So actually taking that light signal and converting it into an electrical signal. And compared it to various outgroup species. Otherwise, a lot of the same methods as I talked about before. So talking about cone and rod phototransduction, so I kind of briefly hinted at this earlier, but essentially it's the same system in rods and cones, but they're paralogous pathways. So they have their own pigments that absorb light, and they have their own intermediaries. So this is called the transducin complex, this is called phosphodiesterase, and here this is a gated channel. And so when these close, that causes hyperpolarization of the cells. So basically we're investigating these genes, genes encoding these proteins, as well as some accessory ones that are associated with restoring the signal. So this is kind of a diagrammatic representation of what I just showed you. So once again, here are the pigments that you would find in a cone. So these are just the cones right here. So pigments you find in the cone, they activate transducin, which activates phosphodiesterase, which activates these channels. And when we look at them in species that we know they have cone function, so things like gorillas and things like marmosets or things like even star-nosed moles, so we conclude from our study that these guys have cones intact. These genes are intact across the board. The one exception you'll see quite often is one of the pigment genes will sometimes be lost. But it doesn't disrupt cone function, it just basically diminishes what kind of colors that an animal can perceive. But when we looked at the armadillo, as well as the sloth, what we notice is a lot of these genes were repeatedly lost. And so you can look at Dasypus right here, the armadillo. At every single stage of this pathway, there were genes inactivated. In the 2-toed sloth, almost every single stage, with the exception of the channels right here. And in fact, the armadillo has the exact same distribution of gene loss as the golden mole right here. So you see all these genes lost, and in fact, I'll shorten the story, so just based off these right here, this gene, the loss of these 2 genes, as these ones as well, suggests that, based off of animal model studies in humans, that armadillos lack cones entirely. And same thing for sloths. Losing this as well as these 2 genes right here abrogates cone function entirely. For Myelodon, since the genome was so fragmentary, we only compared it to the sloth when we could, but we did notice that these genes were inactivated. I'll talk about them in a bit. So here's some examples. You have some actual data as opposed to just a bunch of pretty pictures. So one of the questions, like I said, I wanted to ask is not only were they rod monochromats, which this seemed to suggest that yes, so sloths and at least those 2 sloths and at least that armadillo are rod monochromats, but when did this happen evolutionarily? And I wanted to test the hypothesis that it happened in the most recent common ancestor of these species. So I'll talk about this gene real quick right here that has some shared inactivating mutations. When you have shared inactivating mutations, think of this as a synapomorphy. So if you have the same premature stop codon in the same place in the same gene across multiple species, that seems to suggest that the gene was lost on an earlier branch, so the stem leading to that ground group, right? And so when looking at the armadillo and the sloth, we didn't see any very obvious inactivating mutations like these frame shifts right here or some of these premature stop codons shared between both species. But there was a missense mutation that when we started looking in the literature, we found that when you knock this out in different opsons, it seems to disrupt function entirely. So what I did was, in addition to these species, I did some PCR and sequenced as much of this gene as possible in another 5 armadillos, another 2 sloths, as well as 3 anteaters. And this gene, this cone gene, was inactivated in all of the species that I looked at. And whenever I got this exon, they all shared the same missense mutation, suggesting that this gene was lost in the common ancestor of all xenarthrans. Here's another gene right here. This gene's involved in inactivating the actual cone response, so kind of resetting it back to where it's supposed to be. And what we found is, so here, the 9-banded armadillo, 2-toed sloth, and ground sloth, they all share this massive deletion that spans an exon and an intron, suggesting once again that this gene was lost in the common ancestor of xenarthrans. The thing is, when you lose these 2 genes, even though these are cone specific, it doesn't necessarily disrupt cone function entirely. So we know, for example, I believe mice don't have this gene at all. They're actually able to rescue the function without this gene. And this is just a pigment gene, so that just means that they lost their ability to perceive colors on the stem branch. So there was some vision gene loss happening, but it wasn't full cone loss. And so that's where this gene came in right here. So this is PDE6C. We know from animal models, so you knock this out in mice, you knock it out in zebrafish. If humans have mutations in this that are deleterious, this leads to complete cone dysfunction. And there were 2 premature stop codons that we found that seem to suggest that this might have been lost in the common ancestor. Unfortunately, to me at least, it turns out they were convergent. And so what we think happened is that armadillos and sloths independently inactivated or lost their cones. And so right here you can see these are 4 different sloth species, and they all have premature stop codons right here. Here's some armadillos with some shared frame shift indels. But it's getting at this idea that it probably was lost independently. But nonetheless, we tried to estimate the timing of these gene losses. So here's a nice big phylogeny. So sloths are the sister group to anteaters. And they go way back here about 55 million years ago. And then armadillos are outside of that. Okay? So right to that KPG boundary right here. So what we found is these 2 genes, like I said, were lost in that common ancestor of xenarthrans. So they date back to the Cretaceous, suggesting, once again, a pretty early loss of these genes. Not cone function, but still very early on, these genes were being lost. But then when we dated multiple other genes, it seems like in armadillos, as well as sloths, these genes were lost very early in their history, dating to the Paleocene to Eocene. So kind of keep that in mind, this idea of early loss. But let me kind of get back this idea of cone loss or rod monochromacy in xenarthrans. When we have found rod monochromats, there's only 3 general categories we've actually found in terms of ecological niches. One is in the deep sea. So I talked about whales earlier, so these are very deep diving whales that go to extreme depths, like the sperm whale, for example. And otherwise, in multiple species of fishes, including chondrichthyans as well as teleos, we have found some really deep sea fishes that have lost cones entirely. So you could imagine living in these extreme dim light habitats, very little light except occasional bioluminescence and things like that. Perhaps retaining cones isn't the best thing to do. But beyond that, we've found them in one population of cave salamanders, and otherwise in subterranean species. So the golden moles I mentioned to you guys today, right? So those lack cones. Beyond that, multiple species of caecilians lack cones. And then just this year, there was a study that came out that showed some of these fossorial basal lineages of snakes have also lost cones. So it seems like you only lose cones if you live really, really deep in the ocean, you live in a cave, or you live underground. They have to be really extreme dim light niches. So not just being nocturnal is enough. So that raises the question of why would xenarthrans then lose their cones? That seems very strange if you think about it. This guy right here, this is a fairy armadillo. There's only 2 species of fairy armadillos. And they're extremely subterranean, and so they would fit this description quite well. But beyond that, some species might live in burrows, so like tamanduas. They'll occasionally live in giant armadillo burrows, for example. But tree sloths, they're not living underground, right? They're up in the trees. They're hanging out upside down. Most other armadillos, though they do live in burrows, they hang out outside all the time, in the daytime as well as at night. So why would there be a loss of cones at all? So if you actually look at the fossil record, in terms of how many species there are of fossorial adapted, so digging adapted species, it actually kind of expands. So there's quite a few armadillos. So this is Peltichelus right here. So it seemed to be a very good digger based off of limb proportions. And even ground sloths. So you might not think of them as this, but giant ground sloths. There's some evidence that they created these paleo caves, these very large burrows down in South America. And you can see on these cave walls these scratch marks that correspond very well with their claws. And if you look at their forelimb morphology, you notice this ulna is incredibly wide. The olecranon is quite prominent, allowing for a lot of muscle attachment, very useful for retracting the arm in a digging motion. And then if we go back to the earliest fossils of xenarthrans, they date to the early Eocene. They were very small armadillos, stem armadillos, and their limb proportions suggest that they were diggers as well. So beyond just the extant taxa, if you look at the fossil record, a lot of species appear to do a lot of digging. But we don't have any stems in xenarthrans, unfortunately. I think that'll be a really cool day once we confirm that. But beyond the Eocene, going back to the Paleocene, definitely into the Mesozoic, we don't have any fossil species. But we do have some synapomorphies, so some shared-derived characters of xenarthrans that suggest that these early xenarthrans were in fact really good at digging. So one example actually comes from the names, or where the name Xenarthra comes from. Xen meaning strange, arthra meaning articulation. And if you look at all xenarthrans, with the exception of one tree sloth, I believe, in addition to the typical zygapophyses that are interlocking between different vertebrae, they have these additional xenarthrous processes. And what they're doing is they're helping to stabilize the spine during digging. So when you're actually digging, you're moving your arms back and forth like that, there's a lot of torsional forces that are generated on your lower back. So by actually bracing the spine with these extra processes, which you find across all xenarthrans pretty much, except once again, some tree sloths have lost them over time, it's very useful for having a nice stable body during digging. On top of that, there's a lot of fusion of the spine towards the lumbar and sacral region, a lot of fusion with the hip. And so they have this sort of synsacrum, not too different from what you might find in birds, once again stabilizing the body during digging. On top of that, if you look at the scapula, so the shoulder blades of these various xenarthran species, so these are all armadillos over here. Here's a sloth, and here's some anteaters. And then here we have some comparisons. What you'll notice is a cat and an opossum, they have a single scapular spine. Okay? So the scapular spine is a point of muscle origination which is involved in retracting the humerus. You look at xenarthrans, they have a secondary scapular spine across armadillos. You even see it in tree sloths, you see it in fossil ground sloths, you see it in anteaters. So that allows for more muscle attachment, once again making a lot of much more stronger movement of that arm. Very useful in digging. And this might seem kind of silly at first, but long claws, or really long, strongly curved claws, are also synapomorphic for xenarthrans. So you think about armadillos, they're spending a lot of time digging, looking up grubs, making burrows, so they have these strong claws. Anteaters, they use these really powerful claws to break up termite nests, right? But then there's sloths. And if you think about sloths, you know, they're living in trees. When you're thinking about a species adapted to living in trees, long claws, I assume for most of you, is not the first thing you think about. Like, okay, if I'm going to live in a tree, I need really long claws so I can hang upside down. Usually you have prehensile hands, you have prehensile tails, some very good grasping structures. But tree sloths have these really long claws, and they both, or not both, but both clades basically, they have the same suspensory posture where they hang upside down. Now, if you actually, basically, even though this seems like this might be a synapomorphy for tree sloths, they both live upside down and they have these long claws, we think these were actually acquired independently. So morphologically and molecularly, we're pretty darn certain that these are independent acquisitions of being suspensory arboreal species. And so if you think about it, once again, that's a very odd posture to possess for living in trees. And so it's possible that having all these digging adaptations, their muscles and their claws and whatnot, canalize them and kind of force them to adopt this very similar suspensory posture. So ultimately what I'm suggesting here is this idea of a subterranean bottleneck that very early xenarthrans went through. So you have this very early loss of vision genes, a few vision genes related to cone function, and then you have some more related specifically that knock out cone function entirely very early on in their independent histories. And you have all these various fossorial adaptations that are involved with strong digging. And so what I'm basically predicting with this, that if we ultimately find some stem xenarthrans, they would be presumably very small subterranean species, and that later xenarthrans have kind of radiated into these various niches. And even though they have radiated, there are some limitations, partially perhaps constrained by their overall morphology, being so strongly adapted to digging, as well as not being able to see in the daylight so well. So if you can't really see where you're going, being a top predator is not exactly the best niche to actually be in. Gliding doesn't seem like a great idea. So anyway, in conclusion, basically what I showed with this research is that mammalian vision, it can regress extensively, and this seems to be associated with living in different habitats. And as I showed with xenarthrans, this can ultimately be used to theoretically infer an earlier lifestyle. So looking at these pseudogenes and dating and using shared and activated mutations, you can kind of point back to a light niche occupied by an ancestral species. I just want to acknowledge all these wonderful people and grants and all the different stuff like that. And that's it. If you have any questions, let me know. Did somebody get the lights there? Let's get dim light again. Oh my God.
Great. Questions?
So I was curious about the actual cost of having cones underground, and do you think that can you look at your genetic data and assess the strength of selection? Do you imagine that this happens just through genetic drift, and then it just kind of, since it's not really a disadvantage losing your cones underground, that it just loss of function just kind of accumulates, or do you actually see evidence for selection for loss of these genes?
So it's really difficult at these deep time scales to actually kind of parse out what are the selection pressures happening. Theoretically, so when we actually compare different, at least mammal species, from what I understand, it's the same pretty much with birds and reptiles, etc. But what you notice is you have this sort of topography of rods and cones. And what it seems like happens is, as you become more diurnal, cones become more present. As you become more nocturnal and dim light adapted, rods become more present. And so it seems like, at least initially, that does seem to be adaptive, right? But at what point, that's the question, right? At what point do you have so few cones? Is it better, is this natural selection actually selecting to get rid of them? Or are they just, not particularly helping, not particularly harming the organism, and so they're just lost in drift? That's a good question, and I'm not entirely sure. But when you lose, one thing to keep in mind though is, when you lose cones, you do lose some function. So most species that have reduced their cone population really small, they don't actually perceive color. They've probably lost one of the populations of cones that's responsible for that. But you do generally lose that ability to see in bright light conditions. And so once you kind of lose that, going back out again becomes very difficult. And so you have to imagine there's either a really strong selection pressure, or they've just been living underground for so long that drift has just kind of gotten rid of it entirely. But parsing that out, it's kind of difficult, at least in these cases. Yeah.
So out of your 65 or so genes that you counted involved, how many of those were total loss versus inactivation? I mean, and do you have the same story between, if you were to look at the phylogenetic story between the two of them?
So yeah, in terms of total gene loss, there were very, very few examples that we actually found. And we weren't entirely sure if it was an assembly issue or what. We did some synteny analyses, and it looked like they might have been lost, but it was very few. And so in fact, in most of these cases, and partly that's probably because the adaptations for becoming subterranean in these species, they weren't really that long ago. We're talking 23 to perhaps 40 million years at the most. We know, for example, in turtles, turtles have lost their teeth somewhere in the Triassic. We think that happened. They still have pseudogenes of teeth genes. And so, presumably you could imagine that those genes can be lost over time. But overall, we saw recovered pseudogenes or fully intact genes almost every single case, with I think 2 or 3 exceptions. It was really uncommon. And once again, it might have been assembly error. So, but it's
So you-
painting the same basic story.
So your gene inactivation most of the time is just a stop codon.
It could be a stop codon, it could be a frameshift indel. There's also some splice site mutations that would disrupt splice function. But yeah, you would see those happen quite a lot. Yes.
So you know which ones have been pseudogenized based on looking at the coding sequence, but for the ones that still have an intact coding sequence, do you know whether they're actually expressed in these
No,
animals?
that's a
Okay.
very good question. And so, one thing I'm interested in, and partially, what we're planning on doing for research here, is looking at things like non-coding sequences and how evolution might happen in these cases. So this is actually allows for useful systems if you know where these genes are inactivated, or you know that they're inactivated based off of these frameshift indels and stop codons. Then you can start getting an idea of comparing some non-coding sequences and what are the kinds of mutations you might see in these examples. But in pretty much all these cases here, in every species we're talking about here, we don't really know with gene expression too much. We know, like one example from the naked mole rat, it doesn't express one of the pigment genes, but it does express another pigment gene. It's a question whether they're actually functional, though. That's another issue. But that is definitely something that needs to be explored in more depth. Absolutely. Yeah.
So I guess my question was similar, which is, it seems like, at least in the anteater branch, they don't, I might misremember, but it looked like SWS1 was lost at the stem, and then you don't, and then you still have active LWS1 in the anteaters.
So we weren't able to look at the anteaters. We tried to get the genes for them, but it just didn't work. But with sloths, so sloths have LWS still intact. But it does bring this question up. So, has that gene been lost entirely? Has it been disrupted through a non-coding mutation, or perhaps it's been co-opted, and that's another possibility. So maybe, we think rods, as you probably well know, evolved from cones. So perhaps somehow rods are able to kind of co-opt some of these other
Yeah,
pigments.
because at least in birds and other organisms, it seems like rod and cone doesn't really sort of a morphological category where you can have the same opsin expressed on something that morphologically looks like a rod, and then in like the sister taxa, the same opsin will be expressed in something that looks like a cone. So I guess my question is have people looked at and done the preps of these guys' retinas to look if they have, maybe they have rhodopsin being expressed, but is it like a coney rod?
So a lot of times what people are doing is they're doing some immunohistochemistry and just kind of checking for expression. And what I've noticed is they'll typically go, okay, so LWS, which is one of the cone opsin genes, LWS is being expressed, therefore this must be a cone, right? And so, and once again it gets us at this idea of what's a rod and a cone. For those of you that don't know, we think geckos, for example, there's this idea of transmutation theory where they had an all cone retina, and that converted to an all rod retina, and then somehow that came back to some of these diurnal species, and that became all cones. So it gets at this idea of what's a cone, what's a rod. Like I said, they have paralogous pathways, and so that's one way of distinguishing these different features. But it's possible that, once again, you have sloths that have lost apparently some cone function, but maybe they're using rods and they've somehow adapted in a way that they're using them functionally like cones.
Do we know what their rods look like?
Yeah, we do. So what people, there are histological studies, and so it always gets tricky with histological studies because, as you said, some rods look like cones, some cones look like rods, right? And so people have thought, okay, these guys probably have all rod retinas. But what we did notice, and consistent with that, is the retinal summation of, so the kind of the nerves downstream of that seem to suggest that same kind of pattern. But what genes are actually being expressed, what proteins are present in these, no one's looked at this like hardly at all, and that's a big thing that probably needs to be done with a lot of these things. But at least behaviorally speaking, if you talk about what sloths are doing, they don't seem to be relying on their vision hardly at all. And when you do shine bright light at them, they make their first of all, they don't even have ciliary muscles to adjust their lens, so they've just completely disrupted. That's just gone, period. And then the actual pupil gets super, super tiny, like extremely tiny, like they're trying to get some information out of there. So it's suggestive that cones are not, or something like a cone is not working. But once again, there's all sorts of interesting questions that happen. You lose something that long ago, are you reinventing it? What's happening with that? So, yeah.
You mentioned potential maybe, or there's rescue genes. I guess my question is this a one-way path once you lose your your cones, you don't get them back, or is there a case of any vertebrate really where you have seen cone loss and then somehow they're able to regain it after maybe going subterranean, coming back in from light?
So
or they just go extinct?
yeah, so like I mentioned, it's, you know, originally rods and cones were defined based off of morphology, and so we didn't obviously know about the different genetic pathways, right? And so really early on, there was this thought, like I mentioned earlier, that geckos had an all-cone retina converted to rods, and we see that in a lot of nocturnal species. They look like rods, but then some species came back and became diurnal, and so they have like cone-like things again, right? But genetically, you know, we're actually looking at the pathways beyond that. No one's looked at that particular example. That'd be a great system. I would love to work on that ultimately. But in terms of actually reinventing the same cone mechanism, you know, no one's really looked at that in any way, shape, or form. But it seems just based off comparative anatomy that might be going on. But we think, generally speaking, if you lose these genes, you're not, you know, getting them back. So can they be co-opted in the same way? I mean, that's a good question. And so some of our data seems to suggest that maybe that's going on. Like I said, if you lose that rod gene, in humans, you can't see at night anymore. It's gone, right? And so what are they actually doing with that? And I'm sure it's species specific. Like I mentioned, mice don't even have a particular gene, but a lot of other mammals have it. They're doing different things, but that's a good question ultimately to ask.
Maybe one more.
I think that the whole group was ancestrally subterranean.
Mm-hmm.
Any ideas as to why so few of them are subterranean now?
That's a nice question. I've never thought about. Perhaps. So I mean, one thought. So there's this general idea that in the Mesozoic, you know, mammals were largely secretive, right? And so you have the big scary dinosaurs and other archosaurs living out there. And so a lot of mammals are very small-bodied. We think might have been nocturnal, and being subterranean is perhaps another way that they've actually done this, right? And so perhaps living underground might have been one kind of adaptive niche to kind of stay out of the dinosaur's way, you know, using broad strokes here. But if you notice, the radiation for xenarthrans is right at that KPG extinction, so right when dinosaurs went extinct. And so it's possible that, just like a lot of other mammals, once you kind of got rid of a lot of these competitors/predators, there was a lot of niche radiation that took place. And so possibly it's just some species, you know, re-emerged and kind of went on completely different paths. So you have weird tree sloths that have all these fossorial modifications that they're not using them anymore. But in terms of why species haven't retained them, I mean, that's just a question of, yeah, why don't species retain the same niche over time? Some of them have, clearly, right?
Yeah, it's slightly different in that you've painted a picture where it seems compelling to me anyway. That's hard to go from being fossorial to being non-fossorial.
Mm-hmm.
You lose a lot of things that are important.
Yeah.
And yet the preponderance of transitions in this clay have been precisely in the direction you don't expect.
But keeping in mind that this is extant taxa, and there's very few extant taxa. There's a lot of fossil species that seem to suggest they retain some degree of fossoriality. And we don't know, like for anteaters, the fossil record is extremely spotty. And so everything was very anteater-ish, and there's only very few species. So the point is, there have been a lot of fossorial species. In terms of extant taxa, armadillos, all of them, to some extent, do live underground, do spend some time underground. They're not completely subterranean, though. So, yeah.
Okay, if you have more questions for Chris, you know where to find him. Thank you very much.